Hey everyone, it's Dr. Hinkey again, here for our second lecture on Chapter 1, looking at Section 1.2. In this chapter, we will look at a little bit about how organisms are classified and named, and so we can separate out and organize our exploration of microbes. Let's take a look at the PowerPoint. So Section 1.2 is a systematic approach. Our objectives for this section: define the term taxonomy, describe the current system used to classify microorganisms, including the 3 domains and 4 kingdoms. Describe how microbes are named and how to properly write and type a scientific name. Know how the name can be abbreviated after being properly written for the first time in a document. So taxonomy is just the naming of organisms. How do we give them their scientific names? And it's important that we have a scientific name that's universal. So we'll see scientific names tend to be Latin or Greek names, and that scientific convention is followed by everyone. So the scientific name of an organism is the same scientific name if you are in the United States or the UK or Australia or Germany or Russia or China or Japan or anywhere. It's the scientific name is the designated name. And that's important because we want to have clear communication. There are a lot of common names that we use that other places have the same common name for a different organism. So making sure that we have just one actual name that's interchangeable no matter what country you go to, no matter where you are, that's the name of the organism. That's our importance of the scientific names, the genus and species name. An example of that would be the love bug. Think about the love bug here, like, oh, those two little things that run around with their tails connected. Okay, that's a love bug. Later on, when we talk about Chagas disease, carried by a parasite that's transmitted through the love bug, but it's a different love bug. That's the common name for it in South and Central America, but it's not ours. So we need to know the scientific name to understand what specific organism we're talking about, to have clear communication. And then how we get to those names is through the process of classification, which is how we organize organisms into different groups based on their shared characteristics. So how are they related? And we tend to think of taxonomy, everything's got a name, it's been named, it's been classified. It's sort of this done deal. But interesting, we've always had the ability, and how we've always classified organisms is phenotypically. What are their shared physical characteristics that we can see, that we can measure, that we can compare and say, gee, that looks like this looks like this, this functions like this. And so our classification has been based on appearances, primarily phenotypes. But now we have the technology that's letting us look at a genetic level at the DNA. How similar is the DNA? And the more similar the DNA between organisms is, the more closely related they are. And so that has really caused a big shakeup in taxonomy. There's a lot of exciting stuff going on in taxonomy now. We'll talk a little bit more about that now, about reorganizing and moving things around as we see that appearances aren't always enough to base our relatedness on. In bacteria, in microbiology, where we're primarily talking about bacteria, we have 2 reference manuals that we use a lot. Bergey's Manual of Determinative Bacteriology and Bergey's Manual of Systematic Bacteriology. And these 2 manuals are our standard reference guides for identifying different types of prokaryotes, small-celled organisms without a nucleus. And we can go through and perform a number of phenotypic tests, like the biochemical testing we'll be doing in lab, observation under the microscope, seeing how they grow on different media, and then compare that with the information in the Bergey's manual to help us identify down to genus and species, what an organism is. So taxonomy's had a pretty interesting history. Carl Linnaeus was the first one to develop this method for categorizing plants and animals. This was back in 1758, so a little bit after Van Leeuwenhoek had seen his wee beasties under his microscope, but before Pasteur and Koch and others had made some greater progress, sort of in the middle. His goal, so Carl Linnaeus lived in some cold European place. Might have been the Netherlands or Sweden, somewhere. And he had this idea. He was going to identify and name and categorize every living thing on the planet. Well, where he lived, he could look out and see maybe 5 species of trees, primarily pine trees, pine forests. We could say there's some pines and a few deciduous trees, a few insects that come back every year, same insects. Our main primary megafauna, bears and moose, and so he hypothesized that there were maybe 1,500 species total in the world. And then he did what great researchers do, and he started sending out his students out into the world. They would go on ships that were exploring, on vessels that were trading, and they would make observations and gather organisms and bring them back. And so he kept continuously finding, maybe I need to up my number. This is kind of interesting, but he started that process that we continue to this day and found that he was off by a few orders of magnitude. But he had a very simple phylogeny. It's a very simple tree of life. He said, there are animals and there are plants. That's what exists in the world. But then as we began to learn more, we expanded on that tree of life here and that phylogenetic tree and found, oh, Monera, there are single-celled organisms, because now we're pretty comfortable with microscope use. And they were different than the animals or plants. They had no nucleus. And then we had protists, very simple single-celled organisms that had a nucleus, and then plants and animals. And then we added the 5th kingdom just as recently as 1969. Until this point, thought that fungus were plants. Not at all. Right? So this division has our Monera. These are single-celled organisms that don't have a nucleus. Our protists, single-celled organisms that are single-celled for their whole life, that do have a nucleus. And our plants that get their food through photosynthesis. Our animals, so they are autotrophs, photoautotrophs. Our animals that get their nutrients, their food, by eating other living things, the chemoheterotrophs. Fungus, turns out, are neither one of these. Fungus are saprobes. They release enzymes into the environment around them to break down large organic molecules, dead and decaying organic molecules, and they break them down and absorb the nutrients. So since they are neither photoautotrophs nor chemoheterotrophs, they have their own, they are saprobes. They have a different method of acquiring nutrients. They have their own division now. So as we learn more, we keep making changes to this. As we'll see, our Monera has divided up. We're in the process of reevaluating this group, the protists. So now what our tree of life really looks like is we have 3 main domains. And what domains are sort of the biggest, broadest category of organisms that have general similarities that differ from the other 2 groups. So we have bacteria, which are single-celled organisms that have no nucleus. We have archaea. and we'll talk about archaea a little bit more. Another single-celled organism with no nucleus. And then our eukarya. And these are organisms that have a nucleus. Their cells have a nucleus. Some of these are single-celled, some are multi-celled animals, fungus, plants. So this current life chart phylogenetic tree with 3 domains, 3 big groups of very distinct organisms. The eukaryotes have more in common with each other than they do with archaea or bacteria. And the bacteria have more in common with each other than they do with archaea or eukaryotes. And archaea are most similar to each other, but most different from eukaryotes or bacteria. So our 3 domains had originally been 2. Previously, the bacteria and the archaea were considered prokaryotes. We talk about these names. These 2 groups, prokaryotes, they were 1 because these are all single-celled. They don't have nuclei. And so they were all grouped together until our technology advanced enough that we could look at them on a molecular level. And it turns out some of those similarities were actually quite different. So the archaea and the bacteria used to be separate kingdoms under the domain prokaryote. We still use that term prokaryote to refer to single-celled organisms with no nucleus because that's what that name means. The prefix pro means early or before. It's like the prologue comes before, early in the book, before the story starts. Prokaryotes came early in life, before there was a nucleus. This prefix "u" means true. Karya is Greek for nut or seed. Under the microscope, the nucleus of a cell looks like if I cut a peach in half and opened it, I'd see that seed, that stone in the middle, which is what a eukaryotic cell looks like under a microscope. So a true seed or a true nut in the middle of my cell versus prokaryotes before there was a nucleus. But based on evaluations of the molecular structure of bacteria and archaea, they were divided. So bacteria have a cell wall. Most bacteria have cell walls made of peptidoglycan. Archaea, those with the cell wall, have a different structure, different molecular structure to their cell wall. It's made of pseudo-peptidoglycan, what we call it, to divide those. One component of their ribosomes is actually different from the bacteria and more similar to eukaryotic structures. So they broke off from bacteria in this tree. Right? So our original prokaryotic single-cell, no nucleus organism differentiated and developed into a group that had some structures that differed from bacteria significantly and were more similar to eukaryotes, primarily in those ribosomes. Now, eukaryotes went even further and developed a nucleus and membrane-bound organelles. Neither of these groups has membrane-bound organelles. They have ribosomes, which are organelles, but they don't have a membrane. They can have inclusion bodies, which are little storage containers, but they don't have a membrane around them. Our eukaryotes, as we'll go into more detail in chapter 3, have membrane-bound organelles. And here we have broken down that group that was protists, our single-celled organisms. We've broken them down further now into subgroups based on motility, the ciliates, those that move with cilia, those that move with flagella, those that are immobile in their adult phase. So we've got them broken down further. We have some broken down based on their similarity to how they obtain nutrients. So slime molds are similar to fungus in how they get their nutrients, but they're different from fungus too in their cell wall structure. We have some that move through amoeboid or pseudopods, amoeboid movement. and then our animals. So within any domain, we have the most similarity, but we also have significant differences. So we need to divide even further. But that first big difference is our current phylogenetic tree that is most accurate for our knowledge that we have now. It was developed by Royce and Fox, and they used ribosomal RNA sequences to determine similarities and differences. So if we look at our 8 major taxa, so taxonomy, the naming, our main naming categories from the broadest, so the most inclusive, the domains, all bacteria have similarities, all archaea have similarities, all eukaryotes have similarities. Domain is the most general, so the most inclusive group has the most number of organisms, and within that we're going to see the most variation within those set of single cells, no nucleus, peptidoglycan cell wall, archaea, pseudopeptidoglycan, single cell tends to live in more extreme environments and have some closer similarities at molecular level and function to eukaryotes than to prokaryotes. From there, our domains are divided into kingdoms. With archaea and bacteria, so in the domain bacteria, the first kingdom is bacteria. And within archaea, the first kingdom is archaea. So they used to be prokaryotes, and then their kingdoms were bacteria and archaeobacteria. But now we've taken that archaeobacteria and they are now archaea. They're their own domain, and so their kingdom now is archaea. From there, we divide them into phylums, classes, orders, family, genus, and species. So once we get to the genus and species level, we're at our most specific. We can't just give a species name because the species name is kind of like the first name of us. So my first name is Lynn. There can be lots of Lynns. If I just said, hey, it's Lynn, which one? I don't know. So I'd have to clarify by adding my last name, which would be similar to the genus, Lynn Hinkey. In genus and species, we'd say Hinkey, Lynn. Okay, so within that group. Now I've identified me down to an individual person. So here we're going to identify down to an individual group of organisms or species. This theme begins with the domain, which is our biggest, broadest, most inclusive category based on the cell type, and ends with that most specific group, the species, or the genus and species together. So each domain is divided into a series of kingdoms. Each kingdom made up of several different phyla. Each phylum contains several classes, and on down to our genus and species. So our eukaryotic kingdoms, in our domain of bacteria, the first level kingdom is bacteria. Archaea, the first level is archaea. In the domain eukarya, we have 4 different kingdoms: the plantae, plants; animalia, animals; fungi, our fungus; and protists. And protists is in the process of being divvied up into lots of different groups now that we have the ability to look at a genetic level and put those together. We're going to look at protists in more detail, but that group, that kingdom of protista, was really a catch-all. The only thing those organisms had in common was that they were single cells. Some of them were plant-like and used photosynthesis to acquire food. Some are animal-like, are heterotrophs and eat other organisms. Some are fungus-like the water mold and slime mold, and are saprobes, absorb nutrients from the environment from dead and decaying organic matter. Some are both. Euglena do both. They can either eat other organisms or they can photosynthesize. So they have 2 of those strategies. So it's sort of this catch-all, and there's not really a genetic and evolutionary connection to all the organisms in that kingdom, whereas animals all have an evolutionary connection and common ancestor. Plants have an evolutionary on the same branch, evolutionarily broke off and all have a common ancestor. Fungus broke off, have this common ancestor. Protists, no, kind of all over the place. So now using DNA technology, we can reorder those groups into supergroups. So they're in the process of reevaluating the breakdowns on this. Really, we need to know the 4 kingdoms under the division Eukarya. Our domains are 3 domains: bacteria, archaea, and eukarya. Then our kingdoms of plants, animals, fungus, and protists. And then we'll talk about specific genus and species names when we look at organisms. But a common mnemonic to help remember sort of what the order of these are, should you need it. Dear King Philip came over from Germany swimming. D, domain. A, kingdom. PH, phylum. Class, order, family, genus, species. So we start with domain, the biggest, most inclusive. Then we narrow down until we know exactly what organism we're talking about. So an example of that breakdown, we're looking at the kingdom Animalia. So what domain is that? Everybody said eukaryotes. The cells of all these organisms, sea stars, coral snakes, squirrels, foxes, pandas, black bear, grizzly bear, all of their cells have nuclei. So they are in the domain eukaryote. And they are in the kingdom Animalia. All of these obtain nutrients by eating other organisms. Then we can break down into different phyla. In the phylum Chordata, we're looking at organisms that have a backbone. They're chordates, they have a backbone. So our sea stars are not. Our sea stars are echinoderms. These have their own phylum based on similarities that they have with other echinoderms, like sand dollars and diadema, sea urchins. But they don't have a backbone, so they fall out. We become narrow. We've narrowed down our inclusivity here and eliminated those without a backbone. So sea stars are out. So in our chordata, we're a little narrower. Then we're going to get to the class level. And now we are going to be in the class Mammalia. Our snake is a reptile, that is their class. They fall out. Mammals have in common that they have hair and females produce milk for their young. Snakes don't do that, so they fall off out of this category. Then, out of all of our mammals, we can identify further and get to the order level of carnivores. Now we have carnivores, organisms that eat meat. Well, squirrels do not eat meat, and so they fall out of this category. Then we get to the family level, Ursidae, the bears, and our fox falls out. And then we get to genus Ursus, the polar bears, black bears, grizzly bears are all in the genus Ursus, but panda bears are not. And then if I want to specifically identify the grizzly bear, Ursus arctos. I can't just say Arctos, because that's just a common name. I could use that same species to, say I'm looking at a fox that lives in the Arctic, to name a fox. So species name can be reused, just like there can be lots of lins, there can be lots of organisms with the same species. We'll see the species name Aureus, Aureus, it means gold. Use that species name for a lot of things, a lot of organisms that are yellow in color. So I don't know what Aureus I'm talking about unless I connect it to the genus. So here I don't know what Arctos I'm talking about unless I connect it to a genus. So notice species, I have genus and species, and that identifies my organism. A species is a group of organisms that are capable of reproducing with one another. They're genetically similar and capable of reproducing and having viable offspring. So Ursus arctos is down to my species name. So when we assign scientific names, we use that genus and species. That's the scientific name. We say it's binomial nomenclature. Binomial, by two nomen names, so binomial nomenclature, two names, and Carl Linnaeus is the one who developed this system that we still use to this day. Scientific names always that combination of genus and species. And when we write this out, the genus name is always capitalized and the species name is always lowercase. Both parts are italicized. You may have learned or heard that, oh, you could underline. Underlining was actually a convention for typewriters because typewriters didn't have italics. The font you had on those keys was the font you got. And so other than having a printing press where you could change the font, you couldn't italicize, but you could underline with a typewriter. And so that convention was established when typewriters came about. But now we all have word processors. So the proper way to write a genus and species name is to italicize it. You may notice if you're reading something where all of the font is italicized, then the genus and species name will be just regular font, not italicized. We can still use the convention of underlining if you write by hand and you can't use cursive writing. So you could print everything else and write that in cursive, or if everything's written in cursive or everything's written in print and you can't use cursive, then you can underline the name. We're in college, we all have typewriters and word processors, so it's expected that everything that you submit will be produced on a word processor or on a computer, not handwriting. So you should be able to italicize everything. So some examples of how to properly write the genus and species name. Our genus and species, Homo sapiens. Homo, the genus capitalized. Sapiens, begins with a lowercase, all spelled out. Ursus arctos. We'll look at Staphylococcus aureus, bacteria in the genus Staphylococcus. I mentioned aureus means yellow. See that it can produce a yellow pigment, so it looks yellow when we grow it in the lab. And Escherichia coli, or E. coli. You may have seen that use E. coli quite frequently without establishing that it's Escherichia, because that's a mouthful. But we can only abbreviate like this with just the first letter of the genus name after we've established the genus. So the first time you see it in writing, or the first time you read it, you'll have the full name, genus and species written out. Now I know that I'm talking about Staphylococcus, so I can abbreviate it. So just like if we were writing out my full name, Lynn Hinkey, after we've established that's what Lynn we're talking about, I could now write Lynn H. Only here the genus would be the equivalent to the last name. So we can abbreviate it, but only after you have written out the complete name like this. Now that you all know how to write that name out, you will lose points on every assignment, including those discussion posts, if you don't write it properly. Right, so in our discussion post, there's a question on this. Once you've established this, when you enter your discussion post and open that text box to reply, there's a text editor. The menu comes up across the top. You just have to highlight and hit I to italicize, so there is no reason not to. If you are answering your questions in a Word document or somewhere else and then cutting and pasting them into that text box, go back and proofread because the formatting won't always transfer. So you may have to go back in and re-italicize your genus and species name. But just be aware, now that you know the proper way to do it, I expect you to use the proper way to write genus and species name all the time. All right, let's check how we did. Which of the following is not a domain in the Los and Fox's phylogenetic tree? Plantae, bacteria, archaea, or eukarya. All of these are in the phylogenetic tree, but our domains, we only have 3 domains. Plants fall in the domain eukarya. They are not a domain of their own. They are a kingdom in the domain eukarya. Carl Woese and George Fox sequenced and analyzed what molecule from a wide range of organisms to build their phylogenetic tree. They used ribosomal RNA. And we will learn more about that in chapters 9 and 10 when we talk about genetics. And which of the following is a prokaryotic or microorganism? One thing to bear in mind, learn entire words, not just prefixes, right? The prefix, the base or root word, and the suffix, because pro is early, but our protists also begin with PRO. So don't stop at pro. Make sure you understand prokaryote, before the nut or the seed. No nucleus. Okay, which ones don't have a nucleus? Protista or protozoans is a group within the protista. Protozoan, zoology, animal. Proto, early, but an early animal, so it has a nucleus. They're not in the kingdom animal, they're in the kingdom. But that pro alone isn't going to do it for you. So make sure you analyze the word. Which of the following is a prokaryote? Helminths, bacterium, protozoans, or mold? Molds are fungus. Protozoans, group within the protista, which is in the domain eukarya. Helminths are worms. Bacteria, those are their own domain. They are one of our prokaryotes. Bacteria and archaea are our prokaryotes. So quick check. At this point, everyone should be able to define the term taxonomy, describe the current system used to classify microorganisms, including the 3 domains, bacteria, archaea, and eukarya, and the 4 kingdoms that fall under the eukaryotes: Animalia, Plantae, Fungi, and Protista. Describe how microbes are named. Genus and species, binomial nomenclature, and how to properly write and type a scientific name. And know how the name can be abbreviated after being properly written for the first time in a document. Okay, so that first letter of the genus, always capitalized. First letter of species, always lowercase. So the whole species name is always lowercase. We write out the whole thing the first time we encounter it. And after that, we can abbreviate. Just use capital letter of the genus and then write out the species name, starting with lowercase. All right, that is it for section 1.2. Give your brain a break, walk away, maybe look at some of the other videos, the supplemental videos, take a look at the discussion questions and answer those that pertain to 1.2. And take a look at the PowerPoints and then walk away. Give yourself a little break from this before we come back to section 1.3, looking at the types of microorganisms. That is all for now. I will see you in our next video on the types of microorganisms.